Hello, everybody. Welcome to today's live event. We're going to have the opportunity to learn all about genomics in agriculture and food. My name is Shauna. I am the program coordinator here at Agscape, and here with me today is Dr. Mike Dorrington, who is the manager of strategic partnerships at Ontario Genomics. Today, Mike's going to be sharing some of the history of genomics in agriculture and also be sharing some of the results from case studies that were showcased in a joint project that Agscape did with Ontario Genomics last year. It was called an Exploration of Genomics in Agriculture and Food. And that was launched early last year, and it was also sent to each of you that pre-registered for this event. It is also available in Agscape's digital resource library, which you can find on our website. So if you have any questions about that, don't hesitate to reach out. Before we get started today, please join me for a moment in reflecting about the land that you are joining us from. And we are going to share sort of Agscape's land acknowledgement. And I'll get Mercedes to bring those slides up for me. Agscape acknowledges that we were founded on the traditional territory of many nations, including the Haudenosaunee, the Anishinaabe, the Adirondack, the Mississauga, and Mississaugas of the Credit First Nation, and is now home to many diverse First Nations, Inuit, and Métis people. We are now a fully remote team with many representatives working across the land now called Ontario, and we acknowledge that there are 46 treaties and other agreements that cover the territory. We are thankful to be able to work and live in these territories. We are thankful to the First Nations, Métis, and Inuit people who have cared for these territories since time immemorial and who continue to contribute to the strength of Ontario and to all communities across the province. To continue to center reconciliation, we encourage our team, partners, students, and teachers to take time to learn about the lands they are currently on. Visit native-land.ca for more information. Okay, so we had a little true and false up there. Maybe we'll get Mercedes to pop that back up for us. And then Mike, maybe I'll get you to reveal the answers for us.
Sure. So I'm going to talk a little bit about this in more detail later, but broccoli, cauliflower, and kale are indeed all the same species. And we're going to talk about how they all came to be and what are not related to them, but you might think they are. So
cool. Okay, so let's just jump right into this. We're going to learn all about genomics. Feel free to put your questions for Dr. Mike in the chat during the tour, and we will pop those up, and he'll answer them as he finds a window to do so. And then we'll also have some Q&A at the end. So awesome. Thank you. Take it away, Mike.
Thank you so much, Shauna. It's a real pleasure to be here, and I want to thank all of you for taking the time to join us and to learn more about how studying DNA and living organisms is helping us tackle some of the world's biggest challenges, and especially in the world of agriculture and food production. So if you could go to the next slide, please, Mercedes. Perfect. So before I really get into that, I wanted to talk a little bit about who we are at Ontario Genomics, and as well as people who might be interested in careers in science and in genomics, what it is kind of to work at a place like Ontario Genomics. So what we really are science facilitators. So we're a nonprofit organization that our job is essentially to support genomics research and genomics-based companies across the province. And we do this in a lot of different ways. We help academic researchers access funding for their research. We manage collaborations between university and college scientists with various private industry partners. And we also help provide funds to startup companies in genomics-related fields to help them get going and to help them use kind of new technologies and new ideas to get into the market and to help the world. Now, we also help develop educational resources and training opportunities for people who are looking to work in genomics related fields. And one great example of that is the lesson plans that we put together with Agscape. So if you haven't looked at those, please, I really suggest it. They're fantastic, and Agscape's done a wonderful job in translating some of the research that's happening in Ontario into those lesson plans. Now, our work is really at the boundary of scientific research and business and commercialization. So it's important for people who work at a scientific nonprofit like Ontario Genomics to have kind of a little bit of a background in both. So obviously, it's important that we have a strong scientific background. So for example, I have a PhD in infectious disease research. And worked in laboratories for 10 years, both in Canada and in the US, before coming to OG. But more than just a strong scientific background is the ability to look at what's happening in maybe one scientific discipline and see how that might improve what's happening in another. So, for example, a lot of the kind of big announcements you would have heard about DNA technology and about genomics would probably happen in health, right? But a lot of those things can be applied to agriculture and food, which is part of what I'm going to talk about today. Now, to do all of that, you need to be a good communicator. You need to be a good team player, working with lots of different people from lots of different backgrounds. But most importantly, you have to be curious. You have to be curious about the world around you, about the people that you're interacting with. And you really have to have that drive to be a lifelong learner. So, I was basically in school till I was about 30. So obviously I like learning, and that's been important for my journey, and it allows me to work at a place like OG. So next slide. So today I'm going to talk a little bit about genomics in general. So what is genomics? What do genomicists do? And then we'll talk about how genomics has been applied in agriculture, going back, almost 10,000 years, and then right up till today. And then, as Shauna mentioned, I'm going to talk about some case studies that were brought up in our learning resource that we put together with Agscape. And I'm going to talk about, what kind of happened with some of those research projects, and also where things are going now that those projects have wrapped up. And as Shauna mentioned, we do have a Q&A section at the end here, but I do encourage you to ask your questions throughout, and I'll do my best to answer them. Next slide. Okay, so let's start at the beginning. So what is genomics? Well, at its core, genomics is really just the study of DNA and the DNA molecule. So DNA is essentially the blueprint for all forms of life on Earth. Every living thing, from the smallest bacteria up to the biggest tree, basically has DNA as a set of instructions. It's made up of a 4-letter code that's strung together, and it allows the cells of an organism's body to perform their day-to-day duties, and allows that organism to survive and thrive here on Earth and in whatever environment that it lives in. Now, these genomes, these collections of DNA, have of course evolved greatly over time, and this has been happening over the past couple billion years since life emerged on Earth. And these strings of DNA have expanded and contracted, and they've been cut up and interwoven in different ways. They've been copied and added to and deleted over and over and over again to create the vast array of life that we see around us today. And if we look back at the history of how these genomes have evolved, that gives us information about how they may evolve in the future, as well as how we might use the information encoded in that DNA to perform various tasks. And that's really at the core of what genomics does. And that's where genomicists come in. So next slide. So genomics as a science essentially has come along because new technologies have emerged that allow us to both read and write DNA. So we can look at the DNA of an organism and read that sequence, and through experimentation discover what that sequence is producing in that organism, the traits that that organism might have. So the people who do this are called genomicists, and they analyze genomes in their entirety, as opposed to a geneticist, so in genetics, that might look at single genes or maybe small collections or families of genes and how they're passed on from generation to generation. Genomicists instead will look at the whole genome and how it interacts with other genomes out in the earth, out in the world, essentially. So we might look at the entire genome of a cow. We might look at the entire genome of an insect that lives on that cow and how the genomes of those two organisms have evolved together to create that relationship that they have. And once we do this, we can actually ask some really fundamental questions about life. So how has life evolved over time? How do the differences and similarities between our genomes lead to the differences and similarities that we see between us? So you've probably heard before that we share, something like 95% of our DNA with every other mammal on the planet, right? So that 5% that's different is obviously very important, and genomicists can look at how that occurs. Also, how do genes across the genome work together to create the traits that we see in individuals? And how can we use that information to, for example, improve our health or improve the health of livestock or our pets or even the health of our environment, right? How do we improve the yield of a crop or help it fight off infections, for example? So these are difficult questions, right? This isn't easy science by any stretch of the imagination, but we now have the tools and we've reached a point scientifically where we can start answering lots of those questions. And that's what a genomicist does. So next slide. So our ability to accurately read DNA and certainly to write it has only kind of come about in the past 25 years or so. So the Human Genome Project was kind of the first example of us sequencing or reading an entire genome, the human genome. And in fact, really, if we look deeply, that wasn't really accomplished until like last year of getting the entire human genome. But we have been playing around with DNA for about 10,000 years, and people just didn't know it yet. So around 10,000 years ago, we shifted from being hunter-gatherers primarily to growing our own food, to becoming farmers primarily. And since then, farmers have been working to create new crops. And a great example of this was brought up in the true and false question. So if you look at the slide here, right, in the middle, we have the common wild mustard, or Brassica oleracea. Now, farmers who were cultivating this wild mustard found, some plants had bigger leaves than others. Some plants had more developed stems or deeper roots, or these different traits. And what they discovered is if they bred two plants together that had, for example, big leaves, then their offspring would have big leaves, right? So this was kind of the beginnings of understanding genetics and how traits are passed down through generations. Now, when they selected for those big leafed mustard plants over and over and over and over again, eventually, about 1700 years ago, we started cultivating kale. So kale is just the wild mustard plant that's been selected over and over and over again to have large leaves. Farmers did the same with selection for large roots, and that became turnips. Different buds on the plant were selected for, and they became broccoli or cabbage or cauliflower. And if you looked at the stem and how it evolved over time, we can create kohlrabi based on that. Next slide. And so there's many, many vegetables that have come from this one plant. So here's some other examples: broccolini, romesco, the orange cauliflower, and there's even others: collard greens, Chinese broccoli, mustard greens, Brussels sprouts, all come from that one plant. But we can't just look at a plant and its outward appearance or its physical traits to determine whether they are related to these other plants. So rapini over here looks a lot like kind of a mixture of, I would say, mustard greens and broccolini, but it's actually not related to these other plants. It doesn't come from the wild mustard. So we have to look a little deeper at the DNA sequence. Next slide. So creating these ancient crops, as I've kind of alluded to, took a long time. Hundreds and hundreds of years of careful selection and breeding to lead to these new crops. Now, today, crop scientists can do a very similar process, but because we've gotten rather good at it, we can basically go from one crop to another in about 10 years, as long as that jump isn't too significant. So, for example, scientists could take a corn plant that is resistant to a particular virus, just naturally resistant to it, and cross it with a corn plant that's particularly high yielding, right? They can cross those together to create a plant over a few generations that is both virus resistant and high yielding, right? So we call that conventional breeding or selective breeding. And this is based on the traits that those crops have, right? It's not based on their DNA sequence. Ooh, Autumn Unwin, great question. Is there an experiment kids can do at home to explore genomics? Well, there's a number of experiments, and I'd be happy to share some resources after. There's some great resources out there that deal with this, but there's very simple ways to extract DNA, for example, from a strawberry using basically household detergents and things like that. So yeah, there's lots of stuff that we can explore at home. And yeah, I'll definitely share some resources after this. So trait-based breeding is one thing, right? We can look at two plants, see that they're different, and try to breed them together to get a good combination of those traits. But now that we can actually read the DNA sequence, we can be a lot more selective about this, right? We can actually go into the DNA sequence and see, okay, here is the gene that's leading to that viral resistance, for example. And we can, with essentially molecular scissors, extract that gene and integrate it into another crop varietal. So, for example, that high yielding crop. And in this way, we can be much more selective. And we can take less time and less resources, fewer generations of breeding to get to that crop that is both viral resistant and high yielding. Now we can also do this. So sorry, that's called gene editing. Now we can also do this by taking genes from a completely different organism and put it into, for example, a crop. So that's called genetic modification. So if you've ever heard of GMOs, that's what's happening there, is a gene from a different organism is being put into that crop or whatever it is. And so a great example of that is the Bt toxin. So this is it comes from a bacteria that naturally produces a toxin that doesn't allow a certain type of moth to grow. And these moths can be devastating to crops. And so what scientists have done is put that toxin into the crop so that moths can't eat the crop anymore. And that protects the crop and allows us to use fewer things like insecticides and stuff like that. So very important for the development of new crops and for ensuring that they can grow unencumbered and feed the world. So next slide. So now I'm going to go through a few of these case studies that were brought up in our learning resource. And these are all projects that were collaborations between an academic scientist at a university and a private industry partner, and them working together towards a solution to a problem. And all of these wrapped up in the past 6 months. And all of these happened in Ontario. So this is real science that's happening right now, right here, which I think is really, really exciting. So we'll go to the next slide. But before I get into that, I guess I want to talk about the fourth agricultural revolution. And so there's been these 4 revolutions in agriculture. The first was our movement from being hunter-gatherers to farmers. And then the second was going from just farming to sustain a small group of people to farming for an economic benefit. So exporting and things like that. And then the third was the introduction of more high yield crops, selective breeding, things like that, and the increased mechanization of farming. Now, the fourth here really comes about because of these new genomic and other technologies like artificial intelligence, drones, things like that. Really, farming is being driven by technology now, and that's what this revolution is all about. And it's important because the world is changing, right? We're facing some really, really tricky challenges right now. So by the year 2100, so that's what, 70 something years from now, there will be 3 billion more people on the planet than there are right now. So it's almost a 50% increase, going from 8 to 11 billion people in that time. We need to feed all those people. We need to clothe all those people. We need to make medicine for all those people. And agriculture is absolutely key to all of that. At the same time, climate change is leading to more and more adverse weather events like storms and droughts and things like that, which are making growing crops more difficult. And then there's also market demands. So consumer demands change over time. And so too must the food and beverage producers and processors that we work with. So these projects that I'm going to talk about are all vital to this fourth agricultural revolution and ensuring that we can do all these things. So next slide. Okay, so first I'm going to talk about one of the ways that scientists are working to combat climate change and create more adaptable crops. And this is through something called bioinoculants. Now, many, if not all of you, will be familiar with the microbiome. So these are the collection of tiny organisms, bacteria, viruses, fungi that live on our bodies and inside of us. And these microbiomes are super important, right? We need them in order to properly digest food. Without them, we would be sick with infectious diseases much more often. They even produce things like clotting factors so that we can heal small cuts and things like that. Without these bacteria, we couldn't do any of those things. Now, it's not just animals that have microbiomes. In fact, almost every living organism that you come into contact with has a microbiome, and that includes plants. And the plant microbiome is just as important as an animal microbiome. The plant microbiome allows plants to extract nutrients from the soil, to absorb and retain water efficiently. And again, to prevent infection from other microbes. So basically, these are things that help sustain a plant as it grows. And so scientists are now creating these bioinoculants, which are just bacteria, to help do that even better than the natural microbiome might. And so the goal is to allow crops to adapt to climate change without this long process of having to develop new crops over and over and over again as our world changes. So next slide. So one of the people that we're working with on this is Dr. Neil Emery and his lab, which is at beautiful Trent University in Peterborough. And Dr. Emery's group works on altering the microbiomes of various crops so that they can better grow in our volatile climate. So they study the natural microbiome of these crops and use specialized techniques to determine which bacteria are having the biggest impact or basically providing the most to that plant so it can grow more. And Neil's lab has teamed up with the company NutraAg to test whether a particular bacteria could help crops grow even in drought conditions. So next slide. So what they have found is that if they add a bacterium that's called methylobacteria to various crop seeds as they're being planted, basically those bacteria will support that plant. The plant will grow faster, produce higher yields than plants that do not have this bacteria, this treatment given to their seeds, I guess. And the crops with the bioinoculant, with the methylobacteria, were also more resistant to drought stress and better able to retain water, even when water wasn't being provided. So this is really, really important and has two facets, really, right? Not only will this plant grow better in drought conditions, but even in good conditions, right? Farmers can use less water to grow these plants and hopefully less fertilizer as well. So it's going to create a big economic benefit for the farmer, but also a really big benefit on greenhouse gas emission and water usage and things like that. So Nutrag has already put this on the market, so farmers can use it right away, but the work will still continue. Right? Science never stops, and more bioinoculants are being developed, and methylobacteria will be tested on more crops. So what are the barriers to doing future food genomics work from Mira? Yeah, that's, again, a really important question. And part of the barrier here is, well, there's lots actually. So there's probably not enough funding going into this type of research, and so it's difficult to choose kind of where that funding should go. As I said, the planet is changing extremely rapidly, and so it can be hard to keep up with things. And you know, there might be kind of simpler tools than genomics to try to tackle these, but they might not be as effective. And just, you know, and things like sharing data among scientists and, you know, ensuring that we're all working together and we're not so focused on, you know, patenting things. We're sharing information. If we do this with an eye towards helping, as opposed to, you know, as opposed to benefiting on a personal level, then some of those barriers will be broken down, certainly. And we just need more and more information. We need to sequence more and more crops. We need to sequence more and more bacteria. We need to get more and more information so that we can use that information to make good decisions. So thank you, Mira. Okay, yeah, so that was the end of this slide too. So next slide. Okay, so next is food security. And so as I mentioned, right, 11 billion people in the next 77 years or so. It's gonna be really tough to feed those people, to feed all of those people, right? Especially because of things like climate change and the need to use land for other things. So one of the ways in which food security is being tackled is to try to actually expand the area that certain crops can grow in. So basically adapting crops to new or changing environments. And research on this in Canada, which of course is a cold weather climate, right? Lots of crops won't grow well in Canada because it just gets too cold in the winter, or our winters are too long, or our days are too short. But we can adapt crops to actually thrive in those conditions. And so various crops are being researched in this area, including hazelnuts, wheat, soybeans, and even grapes for wine. I'm from the Niagara region, so very important to our region, of course, is the wine grapes, and we want them to continue to grow here. So next slide. But I'm going to focus on hazelnuts here, and there's a simple reason for that. Well, we have a project with a great hazelnut company in Ferrero, and I know that many of you, if not most of you, love Nutella and other hazelnut-based products. So you'll be happy to know that a lot of those are made here in Ontario. Ferrero is a big processing plant in Brantford. And the problem is that they have to ship these hazelnuts in from elsewhere. Because while there are hazelnuts that grow in Ontario, they're not the right hazelnuts for Ferrero's products. Those hazelnuts grow in warmer climates, places like Italy and Turkey and the Pacific Northwest of the United States. And they have to ship all those hazelnuts in to create the products here in Ontario. Obviously, that's expensive. And, you know, it's also not great for the environment, all the fuel that's needed to get those hazelnuts here. So Ferrero would love if the appropriate hazelnuts could be grown here as well. Now, the problem with that is that the hazelnut fruit, or the nut itself, starts developing in February and March when we're still having sub-zero temperatures overnight. And the hazelnut doesn't like that. It gets stressed out. It sees that it's too cold to reproduce. And so it converts all of its energy into survival instead of reproduction. And so they don't produce the hazelnuts that we need. Next slide. So, sorry, next slide, Mercedes. So Dr. Saxena's lab has discovered that Canadian hazelnut trees have actually evolved to deal with this cold by producing certain chemicals called indolamines. And indolamines are just very simple small molecules. One example that you may have heard of is tryptophan. I'm sure you've heard that that's why you get sleepy after Thanksgiving meal. But tryptophan is one of these indolamines, and in the context of a hazelnut tree, basically prevents the hazelnut tree from getting too stressed. So Canadian hazelnuts produce lots of tryptophan in the late winter and early spring to deal with that stress that comes with getting cold at night. Whereas the Italian hazelnuts have not evolved this stress mechanism. So Dr. Saxena, through his work, has discovered this and now can work with Ferrero to boost indolamine production in the Italian hazelnut trees and hopefully be able to grow them here in Canada, which would reduce the need for shipping them and provide a new crop for farmers here in Ontario, which is very exciting. Now, of course, they're going to look at other crops as well and see how their indolamine pathways work, and hopefully be able to boost production of various crops in cold weather climates like Canada, but also elsewhere in the world. So we're expanding the range of these crops where they can grow. Next slide. So finally, I want to talk about the concept of natural diversity. So when you look around and you see all these different organisms around, you can tell, obviously, there's a lot of diversity between species. There's a lot of diversity between us and a bee or a flower, something like that, right? But there's also a lot of diversity within species, right? You are different than your classmates, right? There's diversity within the human population, and that's true of every single species on the planet. And we can harness that diversity to get organisms to do new things for us. And one of the ways that we've been harnessing a natural process for thousands of years is brewing or fermentation, right? People, again, thousands of years ago discovered that if you allow food to rot in a vessel that's protected from the elements, alcohol will be produced. And people have used that for various different uses over the years. Now, the way that this really happens, and of course they didn't know this back then, was that there are things on that fruit or that grain, whatever, that are single-celled fungi called yeasts, which basically eat the sugar from that fruit or that grain and convert it into energy for itself, and then as a byproduct create carbon dioxide and alcohol. And that's what we're taking advantage of, right? The yeast isn't in there saying, oh, I'm going to make alcohol for these humans, right? It's just a byproduct that we happen to find useful. Next slide. However, Dr. George Vandermeer at the University of Guelph knows that not all yeasts are the same. And in fact, even very similar yeasts that are very closely related, when you look at their genomes, will have different ways of converting that sugar into alcohol and will produce different byproducts that might change the flavor or the aroma of the beer that's being produced, for example. So Dr. Vandermeer has teamed up with a company called Escarpment Laboratories in Guelph, who sell different yeasts to brewers across the country, to create a database of these yeasts, so the genomes of the yeasts, and then what types of byproducts they'll produce, what you can expect, I guess, from a beer that's brewed with that yeast. So it's very important to the craft brewing industry as they try to use different sugar sources, yeasts, or other ingredients to create beers with improved flavor and aroma for beer snobs like myself. Next slide. Now, this is a bit of a confusing diagram. I don't want anybody to worry about that, though. All you need to know is that all the dots around the outside of this circle are different yeasts, but they all belong to the same species. These are all brewer's yeasts, which are the species Saccharomyces cerevisiae, but they are all slightly different in their genomes. Okay, so Dr. Vandermeer basically went about and sequenced or read the DNA sequence of over 20 different beer yeasts, and then looked at those genomes, and then also looked at the flavor compounds that they were producing, or the temperature that they best like to work at, to ferment at, or the amount of time it takes for them to consume sugar, or their preferred sugar source, all these different traits that these different yeasts have, and compared that to their genomes. So that allows us, A, to be able to take those individual yeasts and say, here's a yeast that will produce maybe a fruity flavor or a funky smell that people might like, right? But also what it allows us to do is if we take a wild yeast, a completely random yeast out of the wild, we can then sequence the genome of that yeast and compare it to the yeast that we already know about and get an idea of what it will do if we try to use it for brewing. And this doesn't stop with beer, of course. We can do this with yeasts that are used to create things like sake or bread, or even to ferment grapes into wine. And perhaps even most excitingly, this is not just about creating alcohol. In fact, some bacteria and yeasts in the wild will take in sugars, and instead of creating alcohol and carbon dioxide, will create plastic. So biodegradable plastics that we could use instead of the plastics that we now derive from fossil fuels, for example. We can also engineer, because we can write DNA as well, we can engineer yeasts to take in new food sources, things like food waste, from your green bin, for example. We can convert that into things using these engineered yeasts. We can make things like textiles, like nylon, or fuels for powering equipment, or even jet fuel, or even food ingredients like gelatin or vitamins, things like that. So the power of biology and the power of DNA, which is found in all living things, right, has the potential to change how we produce just about everything, and can do so in a way that is less destructive to the environment, while also ensuring that everybody has what they need. Ooh, another question from Mira. Perfect. What is important for people to know about GMOs? They can have a bad name, even though we know they're misunderstood, certainly. So the first thing I'm going to say is, if you haven't taken a look at the resources that we've produced with Agscape, there is some great resources in there as far as debunking some of the myths around GMOs. I would say the most important thing, though, that I'll mention here today is no one is taking a gene, putting it in a crop, and growing it in a field and growing food for someone right away. There is a lengthy and a lengthy process and regulations involved in going from one plant, putting a gene in, and then having that served to the public. There's a ton of work, a ton of research, and assurances that these things are safe, things before they come to market. And the other really important thing is that, again, we are not going to be able to feed 11 billion people on this planet and provide them with the nutrition that they need, not just quantity, but quality, without the use of GMOs. And we can support the health of these tens of billions of people through agriculture and through our own inventiveness and innovation. And so while there needs to be those backstops, those regulations to ensure that we're being safe, places like Canada are at the forefront of ensuring that, while providing the world with the food we need, with the textiles we need, with the medicines we need. And these are all coming from nature still, right? Even though if we're mixing genomes together, it's not random, it's very targeted, and very safe. So again, take a look at those resources that we've put together. There's some great debunking facts about GMOs in there, and share that information far and wide, because there are people who don't understand the technology, and that scares them, and I'm fully supportive of that. But the more we learn, the more we know that these are very important for our future as a species, and for the future of our planet. How did I get into studying genomics? What pathways did I have to study to get where I am now? Okay, so that's a very, very cool question, Autumn. And one of the things that I tried to mention at the very beginning is genomics really impacts all areas of the life sciences. So health, agriculture, clean technologies, all these different things, and it's very interdisciplinary. It's very cross-sectoral. So there is no one way, I would say, of getting into this. So for me personally, as I mentioned, I was an infectious disease researcher, and I studied the interaction between our genome, the human genome, and the genomes of pathogenic bacteria. And actually, they were opportunistic bacteria. So ones that weren't normally able to infect us, but as soon as we had another infection or became compromised in some way, then these tend to spring up. And so there's very important kind of interactions between our genome and the genome of those bacteria. And so that's how I really got into the study of genomics. But there's plenty of ways. You could become a crop scientist or a food scientist. You could work in health. You could look at these microbes I talked about that make plastic, for example. How do we make them do that more efficiently? There's the possibilities with genomics are endless, and therefore kind of the ways into it are endless as well. So what I would say is, if you're interested in genomics, reach out to us. We love talking to people about genomics and the power of genomics. But also, think about a problem or a challenge that you see in our society or on our planet, right? And then think about all the ways that living organisms can contribute to us solving those challenges, or tackling those challenges, or solving those problems. And you'll find that there are lots of people working on those types of things, and there's so much information out there, and there's also so much to learn. One of the things that we try to get across is a scientific project cannot live by itself, right? There's a bunch of stuff that came before, and there's a bunch of stuff that's going to come after. It's all part of one big process. So don't feel like we figured any of this out to completion. There's always new questions to be asked, and there's always new things to be learned. And it's really exciting to be at the forefront of that, and to again be that kind of lifelong learner. So I think that's my last slide there. If you want to go to the next slide, Mercedes, or we can, no, it just says questions. So if there are any other questions, feel free to ask. Also, feel free to reach out to me. That is actually the wrong email address. There's no G after the M, but Agscape can help you get in contact with me. That's my mistake. And yeah, I'd love to talk to you all more about these different projects and different ways that we're going about this. So how can normal consumer youth support the growth and awareness of genomics, especially within agriculture? Yeah, great question. So what I would say is, when I took this job, one of the things that people said to me is, well, you'll be the most interesting person at any dinner party now, because there's millions of questions to be asked, and we get kind of a front row seat to what's being asked, what's being developed, what research is going on, right? So the most important thing is finding out what the people around you are interested in, and then talking to them about these cool things that are happening around them that they might not know about, right? Again, I come from a health background. My research didn't look at agriculture or food production or anything like that at all. And yet almost everything that I learned in that health space has been applicable to me learning about agriculture and food production. So don't feel like you don't know enough, but be curious, ask questions of people, and start these conversations. We're never going to be comfortable with how these new technologies are affecting our lives if we don't understand them at any level. So I would say, do your best to learn. Again, these resources are a great start that we put together with Agscape. And if you have other questions, then don't be afraid to ask, and involve other people in those conversations.
Well, thank you so much, Mike. That I every time we talk, I learn more and more and more from you. And I just, as a teacher, I'm just always so inspired. When the wheels get turned, like, oh, we could have a conversation about this, or we could go down this rabbit hole. And it's always just so much fun learning from you. So I'm really glad you were able to take the time today to share with us. And
it's my absolute pleasure. And like I said, this This is one of the most fun parts of my job. So if people have other questions or you want to learn more, then just reach out and we're happy to help.
Wonderful. Oh, yeah. People are saying thank you in the chat as well. Well, happy Canadian Agricultural Literacy Month to everyone. Thank you for joining us throughout the month on all of our little field trips and virtual experiences. I know we've touched on lots of really cool topics this month, so we're super grateful that everyone could join us today, especially you, Dr. Mike. And yeah, thanks a lot and take care, everybody.
Awesome. See you, everybody.